This is Bio 181, Chapter 10, which is on mitosis and how cells divide. So we're going to start generally and just give a quick shout out to bacteria, and then we're going to leave them in the dust. These poor little cells don't get much attention in this course, but bacterial cells, if you remember, those are single-celled organisms. They're prokaryotic, so all prokaryotic cells are going to reproduce this way by and large. So you have a single cell, and remember, in a bacterial cell, they just have a single chromosome of DNA in the center. The characteristics of a cell that are common to every single cell are the fact that it has, I'm just going to use this to revisit it really quick. So a cell membrane, cell membrane, it's going to change the color there. All right, cell membrane, plasma membrane, phospholipid bilayer, DNA inside. It's got some ribosomes in here, and then it's got this other stuff called cytoplasm. So every cell has those four characteristics. Now, bacterial cells, they're very simple. They reproduce asexually, which means they're not changing their genetic code by mixing it up when they're reproducing. So the method is binary fission. Binary, we're going to split it into two, and we're going to make exact copies. So the DNA is replicated. It's going to be the exact same DNA in both of the daughter cells, right? The same exact genetics and identical ribosomes. And there you have it. That's basically it for bacteria. Okay, they, of course, would have these as well. Same thing. So they're clones, making exactly the same offspring. In eukaryotes, we're going to talk about how they reproduce for the rest of this chapter 10, and chapter 11. So we're going to start off by talking about eukaryotes. Now, remember that eukaryotes, by definition, they are, oh gosh, trying to get this. There we go. Okay, they are cells that have a nucleus. So their DNA is located inside of a membrane-bound compartment, and then in there, they've got multiple linear chromosomes in the nucleus. So multiple linear chromosomes, whereas the bacteria or the prokaryotes had just one single circular chromosome. The chromosome is a combination of proteins and DNA. So remember, DNA, that's our genetic code, right? It stores genetic information. So that's what this is, right? And then the proteins are really to help package it. So these are here to help bind and package and store that genetic information. So a few more words. We've got a lot of vocabulary in this chapter. I'm going to go through it fast. So haploid means you have one set of chromosomes. They're all different genes in different orders. Diploid is you have two sets of chromosomes. So each set is a sequence where you have on both sets, you have so one of each that's going to have genes with the same sequence. They might have slightly different versions of the gene, but you have two sets of each version. So in humans, we have two sets. We've got one from our mom. So the mom gives us 23 chromosomes. The dad gives us 23 chromosomes. And so we have a total of 46 chromosomes. So like a haploid cell, just to back up, so one set, so the egg that's produced in a human is haploid. And so that would only have 23 chromosomes. Also, the sperm is haploid, and that's also going to have only 23 chromosomes. So when they combine the egg and the sperm, then you get a full human being, you get a zygote, which develops into an embryo. All of those cells are going to have 46 chromosomes. All right, so that's for humans. It's going to vary the number, with different organisms. Homologous chromosome, that refers to this set of chromosomes that have the same sequence of genes. And I know I didn't define genes yet, but a gene is basically a sequence of DNA. So this is a sequence of DNA that codes for a protein, that codes for a specific protein. Or I'll just, you know what, I'm not going to say specific. I'm going to say for a protein, because there's some nuance to that. All right. So that's homologous. So a homologous pair, those are going to be like the number one chromosome from your mom, the number one chromosome from your dad. Sister chromatids, these are, so sister chromatids, okay. It's similar word to chromosome, but chromatid. Now, chromatid, these are identical. So in this case, I'm going to get a different color here because I'm tired of seeing all this red. So you've got one chromosome here. The exact same information is duplicated and connected to the other sister chromatid. So they're connected and they're identical. So they're sisters. They're basically identical sisters. So these two sequences are identical. Identical chromosomes. But the trick is, so I really can't say chromosome because together, when they're hooked together, they're called one chromosome. So it's kind of odd, but as long as it's hooked together, it's called one chromosome. So, but each of the sister chromatids has an identical sequence of DNA. All right. I don't know why it switches to red there. The last thing is centromere in terms of this vocabulary. So you've got you've got a chromosome here. They're connected. That center part where they're connected is called the centromere. All right. Okay, cell cycle. The cell cycle is basically the life cycle of a single cell. So from the time that the cell divides and it's brand new until it's going to divide again. So going through this quickly. So we start up here, we got a brand new cell, and then we're going to go through a whole cycle where we end up, that cell is going to divide into two daughter cells. Okay, so that's the cell cycle, the cycle of life for a cell. Most of the cycle is going to be interphase. So interphase is going to be the vast majority of the time for most of the cell. So over here, we've got, say, interphase, this long, big phase, maybe here, okay? So most of the cell cycle is going to be interphase. Interphase is broken up into gap 1, which we call G1, synthesis, and gap 2, which is G2. So I'm just going to divide this part here, interphase, into G1, S for synthesis, and G2. So basically, the gap phase, this is when the cell is going to grow, okay? And this is when the cell is going to grow here, right? And it's going to just perform basic maintenance, basic function, okay? So it may not always be growing a lot, but it could grow during that time, and then basic metabolic function of the cell, whatever that cell is made to do. So if it's a muscle cell, it's contracting. If it's a gland cell, it's secreting something, okay? So basic function, okay? All right, now, S. S is for synthesis. This is synthesis of DNA. So this is where we make our extra copies of our DNA. So this is where we make the sister chromatids. So the DNA comes in, You have multiple linear chromosomes. I'm just going to write them up here. So say I have, I'm going to make them different sizes. Here's a big sequence of DNA. Say I've got 3 different sequences. So there's the DNA. During synthesis phase, each of those is going to be replicated and hooked to the parent. So it kind of looks like an X. So now we've got that one replicated. That's the long one. Then there's a shorter one, which is going to be replicated and hooked together. And then there's just a little one here with less genes on it hooked together. So now this DNA has been replicated. If you were to follow it through, we'll get to that later. I'm not going to jump ahead. So that's interphase. M phase mitosis, that's going to be where the cell divides the nuclear contents. So nuclear division is mitosis or M phase. That's going to be nuclear division. Nuclear division. Although in common language, when we talk about it, often mitosis will refer to the entire process of cellular division, but technically it's only division of the nucleus. So technically it's only division of the nuclear contents. So that means only the chromosomes are going to be dividing. So in mitosis, that's where you have the contents. Each of the daughter cells is going to end up with 3 chromosomes, just like the parent, identical. So they're identical copies. And anything else I want to say about it? Oh, sometimes the cells can just rest. So they don't go through this phase. They kind of go over here on the side and they rest in a phase G0. And so that's going to be where they are no longer going to be dividing. So they'll just hang out. All right, that's it for this video.